Hello friends, welcome to readmedprep Academy channel. Today in genetics, in Principles of Inheritance and Variation part 4, we shall discuss about karyotyping and pedigree analysis. What is meant by karyotyping? Karyotyping is a technique through which the complete set of chromosomes is separated from a cell and the chromosomes are arranged in pairs. Here you can see the human chromosome which is arranged in pairs from the autosomes as well as the allosomes. The autosomes are 22 pairs and the allosomes is a one pair. In the males XY and in the females it is XX. What is ideogram? An ideogram refers to a diagrammatic representation of the chromosomes. The chromosome morphology, characteristic of a species or a population. This represents an ideogram. What is karyotype? Particular chromosome complement of an individual or a related group of individuals as defined by the chromosome, size, morphology or the number is known as a karyotype. So what is karyotype? Particular chromosome complement of an individual or a related group of individuals as defined by the chromosome, size, morphology and number is known as a karyotype. So karyotype defines the size of the chromosome, positions of the centromere, the presence of secondary constrictions and the size of the satellites. Now preparation of karyotype was done by Tigeo and Levan in 1960. Described a simple method of culturing lymphocytes from the human blood. How mitosis is induced? Mitosis is induced followed by addition of colchicine to arrest the cell division at the metaphase stage and suitable spread of metaphase chromosomes is photographed. The individual chromosomes are cut from the photograph and are arranged in an orderly fashion in homologous pairs. So what is the procedure of karyotyping? About 5 ml of blood is taken and phytohemagglutinin is added to the culture medium of the blood and the blood is incubated at 37 degrees centigrade for 3 days and colchicine is added and the hypotonic saline and the cells are fixed. The cells are spread on the slide by dropping and the cells are digested with trypsin and stained with GM stain and they are analyzed under the metaphase spread and the chromosomes are arranged in a karyotype pattern. What is the use of karyotype? This arrangement of chromosomes in pairs is called karyotype. The chromosome banding permits structural definitions and differentiations of the chromosomes. So what is the application of karyotyping? It helps in gender identification. That is the males having X and Y chromosome, the female having X and X chromosome, that is 2X chromosome. It is used to detect the chromosomal aberrations like deletion. For example, you can see on the right side, the deleted part of the long arm of the chromosome results in a shortening of that arm. So before deletion, the chromosome is normal. After deletion, that arm becomes shortened. Example is 13q deletion syndrome, which occurs in 1 in 5 lakh births. So 46 chromosomes plus XY or XX and the 13th chromosome is deleted of the lower arm. So the children that are born are mentally retarded, have deformed face, no thumbs, heart disease and the short lifespan. The karyotype is shown on the right side. Now chromosomal duplication. A part of the chromosome in the arm becomes duplicated or the genes become doubled. So the arm becomes longer. The duplication occurs resulting in extra genetic material known as human disorders include Charcot-Marie-Tooth disease type 1A, which may be caused by duplication of the gene coding peripheral myelin protein 22 PMP22 on chromosome 17. Now chromosomal translocation. You can see this example, the chromosome number 4 being translocated to chromosome number 20 and a part of the chromosome number 20 being translocated to chromosome number 4. Example of this translocation is of the MYC gene on chromosome 8. The normal MYC gene control growth and division. Translocated MYC genes don't function properly. They lead to cancer of the lymph nodes, otherwise called Burkitt lymphoma. Now, non-disjunction of the chromosomes can also be studied by karyotyping. You can see on the left side the normal separation of the chromosomes during meiotic divisions. Whereas in the center you can see that in the first meiotic division there is non-disjunction of the chromosomes. So the resulting cells will have one part of the chromosome and 3 chromosomes go to another cell. Similarly, the right side you can see the abnormal disjunction in the second meiotic division, which results in the formation of one chromosome in one cell, another part of the chromosome in another cell, and 3 chromosomes in one cell and 3 chromosomes in the another cell. So this non-disjunction results in disorders. Here you can see that during the metaphase the separation of the chromosome occurs by the formation of the microtubules. During the anaphase the separation occurs, but when the separation is in a proper manner, equal half of the chromosomes go on either side of the cell, whereas in a non-disjunction, unequal number of chromosomes go to the different type of cells on either side. Example of this is Klinefelter syndrome. 1 in 1100 births are caused and the chromosome number is 47. They have XXY chromosome and they may also be caused by 23 trisomy. Non-disjunction can also occur. So what are the symptoms? Scarce beard, long fingers and arms, sterile, delicate skin, low mental ability and normal lifespan can occur. It also helps to identify the abnormalities of the chromosomes like aneuploidy. So what is aneuploidy? Where the offsprings that are formed have 2n plus 1 chromosome or 2n minus 1 chromosome. This results because of the non-disjunction during meiosis. So a diploid cell due to the non-disjunction can produce n plus 1 or n minus 1 gametes. On fusion with the other gamete can cause 2n plus 1 or 2n minus 1 resultant diploid cell. So the alteration in the number of the chromosomes is called aneuploidy. So the examples of this is Down syndrome, Patau syndrome and Edwards syndrome. Down syndrome is trisomy 21, Patau syndrome is trisomy 13 and Edwards syndrome is trisomy 18. It also helps in predicting the evolutionary relationship between the different species. Here you can see the relationship of different animals of the Mammalia kingdom by the karyotyping method. Genetic diseases of human beings can also be detected by the karyotyping technique. So disorders caused by individual genes. These changes affect the specific proteins that do important jobs. Incidence of various genetic diseases, for example, phenylketonuria, which is a recessive allele. It occurs in 1 in 60 in Caucasians. Tay-Sachs disease, which is a recessive allele, 1 in 13 Eastern European Jews. Sickle cell anemia, which is a codominant, 1 by 13 in Africans and African Americans. Cystic fibrosis, which is a recessive allele, 1 in 30 in Northern Europeans. Huntington's chorea, which is a dominant allele, 1 in 1000 in people of English descent. Achondroplasia, which is dominant allele, it's a hotspot mutation, 1 in 15,000 in Caucasians. Progeria, which is a recessive allele, 1 in 1 lakh in Caucasians. ALS, which is a recessive allele, 1 in 600 in Caucasians. So, human karyotype can be classified according to the presence of the centromere. So, depending upon the position of the centromere, the chromosomes can be classified based on the 2-arm length. So, there are 4 types of chromosomes based on the position of the centromere. One is metacentric, the other is subcentric, the other is acrocentric, the other one is telocentric. In telocentric, the centromere is at the terminal end. In acrocentric, the centromere is just below the terminal end. In submetacentric, the centromere is just in position away from the center, whereas in metacentric, the centromere is at the center. Grouping of chromosomes. The photograph of chromosomes are arranged in the order of descending length in groups from A to G. You can see that 1, 2, 3 are A chromosomes, 4, 5 are B chromosomes, and 6 to 12 are C chromosomes. And 13, 14, 15 are D chromosomes, 16, 17, 18 are E chromosomes, 19, 20 are F chromosomes, and 21, 22 and the Y chromosome is the G chromosome. So let us talk about the different groups. Group A, chromosome numbers 1, 2, 3. What are the characteristics? They are long metacentric. Chromosome number 2 is submetacentric. In group B, the chromosomes are 4 and 5, fairly long, submetacentric. In group C, the chromosome numbers are 6 to 12 and the X chromosome belong to this group. It's a medium long submetacentric. In group D, the chromosome numbers are 13, 14, 15. The medium long chromosomes acrocentric with a satellite body is present. In group E, the chromosome numbers are 16, 17 and 18. They are fairly short, submetacentric. In group F, the chromosome numbers are 19 and 20. They are short, metacentric. In group G, the chromosome numbers are 21 and 22. They are very short and the satellite body is also present. Now, what is this pedigree analysis? Pedigree is a family tree drawn with genetic symbols, showing the inheritance pathway for specific phenotypic characters. You can see the symbol on the left side and the explanation on the right side. When it is represented by a square, it's a male. When it is represented by a circle, it is a female. When the mating is shown, the male and the female, a line is drawn between them. The parents and children, one boy and a girl, are represented, where one square and a circle are joined by a line, and the children are one square and a circle. In dizygotic twins, which are a male and a female. In a monozygotic twins, a male and a male is predicted here. In sex, which is unspecified, it is shown by a diamond. When there are number of children that are produced, you can see represented by the second child and the third child. Now, you can see here the affected individuals. The affected individuals are shown by colored square box, it's a male, and a colored circle, which is a female. In heterozygotes for autosomal recessive, half is colored, half is non-colored. It's a male, half is colored and half is non-colored, circle in a female. Now, in a carrier of sex-linked recessives, you have a dark circle in the center surrounded by a light circle. In the case of death, the square is slashed by a line. In an abortion or stillbirth, you can see the circle with a small strap above. In a propositus or a proband, you can see a colored square with an arrow. The method of identifying persons in a pedigree here, the propositus in child 2 in generation 2 or the second 2. What is propositus? Propositus means the person from which the generation starts. What is meant by pedigree analysis? Pedigree analysis is the study of traits as they have appeared in a given family line for several past generations. So what is the importance of pedigree analysis? The analysis of inheritance in human families, a very important tool for studying human inherited diseases, allows inferences concerning genotypes in a family or a population, allows predictions concerning phenotypes of offsprings inheriting a genetic disease. So it will be helpful in genetic counseling. Typically small number of offspring, Mendelian ratios rarely observed. This means the normal 3:1 dominant to recessive ratio doesn't usually occur. So today we discuss in genetics in principles of inheritance and variation part 4, karyotyping and pedigree analysis. Thank you very much. Kindly subscribe, like, share and comment the channel Read MedPrep Academy. Log on to readmedprepacademy.com. Facebook ID is readmedprep academy. Our email is readmedprepacademy@gmail.com. Please post your questions in the comment box. We will reply with appropriate answers. Thank you very much.